Hello, everyone. Today we are here to present our project OxyDock. This cutting-edge patented platform represents a leap forward in simulating hypoxic tumor microenvironments and revolutionizing targeted therapy predictions. Let's dive into the innovation that is OxyDock. Cancer, as we know it, thrives on complexity. One such complexity is hypoxia, a state of reduced oxygen levels that can reach from 2% to as low as 0.02%. Under hypoxia, tumor cells become highly resistant to therapies and create acidic microenvironments, as outlined by Hoplin et al. In 2021. These conditions promote genetic instability and aggressive tumor behavior, making hypoxia one of the more formidable challenges in oncology. Targeted therapy is designed to exploit specific biomarkers struggle against these challenges due to the altered biochemical and physical property of tumor cells in hypoxia. To understand this challenge better, let's explore the structural changes induced by hypoxia. So under hypoxia, proteins experience post-translational modifications, such as methylation, phosphorylation, acetylation, and ubiquitination, which significantly alter receptor function. Phosphorylation, for instance, activates pathways during cancer growth, driving cancer growth and survival, while methylation and acetylation modify gene expression to help tumors adapt. These changes create a complex tumor microenvironment that traditional docking environments sadly cannot accurately simulate. So unlocking how drugs bind to target is vital for advancing treatments, and molecular docking such as AutoDock Vina make this possible. So AutoDock Vina, developed by Trott and Nelson in 2010, is a remarkable tool that has revolutionized molecular docking. It can screen up to 10,000 compounds daily, saving around $5,000 in experimental costs. However, it does have its limitations. Most notably, it struggles with simulating hypoxic environments and incorporating post-translational modifications. So an example of AutoDock's limitation is shown with the drug Remorphonet, a drug commonly used in cancers such as melanoma. And as shown here, the experimental IC50 value, or the concentration of drugs needed to kill 50% of cells in vivo, is around 9.75 micromolar under hypoxia conditions. And using a validated equation, as shown here, we converted the IC50 value into a binding energy of -7 kilocalories per mole. However, there is a clear difference when comparing it to AutoDoc's report of a -11.25 kilocalories per mole, signifying the need for in silico studies to accurately represent hypoxic environments. So the solution we propose is OxyDock. Now let's get into the goals and features. Our research has three primary objectives. Number one is to investigate how oxygen levels influence protein ligand binding and post-translational modifications. Number two, validate these predictions against biological outcomes using IC50 data. And number three, develop OxyDock, a novel platform that stimulates hypoxic tumor microenvironments with unparalleled precision while remaining user-friendly for researchers and clinicians alike.
OxyDock is an innovative platform that integrates molecular docking with XGBoost to improve binding affinity predictions under varying oxygen concentrations, crucial for simulating tumor microenvironments. Its user-friendly GUI allows researchers to upload PDB or SDF files, enabling accurate simulations of hypoxic conditions. Unlike traditional tools, OxyDock incorporates environmental variability, which refines predictions through machine learning to address limitations in existing docking methods. To understand its inner workings, let's break down the architecture. So this flowchart outlines OxyDock's workflow, illustrating the integration of parameters, molecular docking, and machine learning. The process begins with the OxyDock GUI, where the users input parameters like oxygen concentration and the PDB SDF files, which are essential for docking simulations. The PDBs are then processed through OxyDock to calculate initial binding affinity, and the results are enhanced using XGBoost to refine predictions. The final outputs, including optimized binding affinities and the visualized ligand and receptor, are then displayed through the GUI. But speaking of XGBoost, let's dive into why we chose this model. XGBoost is a machine learning algorithm known for high predictive accuracy and efficiency, ideal for modeling complex ligand receptor interactions. It handles nonlinear relationships with decision trees and provides feature importance insights. Its sequential tree building minimizes errors, optimizing predictions for time-sensitive situations. The flowchart illustrates how XGBoost builds multiple decision trees sequentially, minimizing residual errors at each step to optimize predictive performance. With this foundation in place, let's demonstrate OxyDock in action. On the screen, you can see the main input section, where the ligand, receptor, and configuration files are uploaded. To the right of that, the molecule visualization panel displays 3D structures of the ligand and receptor and binding. In the output section, OxyDock processes the input data and refines the binding affinity prediction using XGBoost. And in this example, you can see the predicted binding affinity.
So as a first step toward validating OxyDock, we conducted an extensive search in PubMed for targeted therapies in hypoxic conditions, finally narrowing it down to 17 studies. We collected drug information, oxygen conditions, temperature, and IC50 value, which is the concentration in drugs which 50% of cells are dead. And from this, we calculated the binding energy using the Chang-Prusov equation as shown above. So with validation complete, we analyzed OxyDock's position in capturing binding affinities changes across oxygen levels. This clustered bar graph compares AutoDock, experimental, and OxyDock binding affinities under normoxic on the left and hypoxic on the right. Experimental data shows a 50% drop in binding affinity from 21% to 1% oxygen, closely matched by OxyDock's 13% drop. However, AutoDock's predictions deviate significantly with an average 28% error under hypoxic conditions. For example, vermorfinab's experimental binding affinity shifts from negative 8.2 kilocalories per mole to negative 7 kilocalories per mole when comparing hypoxia and normoxia. And OxyDock mirrors this change with a prediction of negative 8.1 to a negative 6.1 kilocalories per mole, respectively, while AutoDock remains unchanged, underscoring its ability to model environmental factors. We also found outliers where binding is better in hypoxia than normoxia, as shown with EGR inhibitor erlotinib, which works better in hypoxia conditions to an alternate HRE-activated EGFR pathway. We can see that OxyDock scoring function has the ability to capture these outliers due to XCBreach's capabilities.
Compared to AutoDock Vina, which has an RMSC of 2.85, OxyDock achieves an RMSC of only 1.16, which is a significant improvement. This accuracy is crucial for developing therapies in hypoxic environments, where slight variations in binding affinity can mean the difference between success and failure in a drug. By significantly reducing prediction errors, OxyDock sets a new standard for molecular docking, paving the way for more reliable drug development. But what does the future hold for OxyDock? Well, the future directions for OxyDock include incorporating less common post-translational modifications for a broader scope of receptor-ligand interactions, collaborating with researchers to validate additional protein-ligand interactions across diverse tumor types, expanding into drug repurposing simulations, testing candidate drugs on cell lines and in vivo models, optimizing pre-processing to make the platform even faster and more user-friendly, enabling widespread adoption. And these advancements will ensure that OxyDock remains at the forefront of molecular docking technology, driving innovation in future research. Finally, let's reflect on OxyDock's impact and potential. To conclude, OxyDock addresses critical gaps in molecular docking by modeling receptor conformal changes specific to hypoxic environments. Its superior accuracy with an RMSE of 1.16 sets it apart from leading models like AutoDock Vina. By enhancing targeted therapy predictions in hypoxic tumor environments, OxyDock paves the way for groundbreaking therapeutic discoveries, bringing us closer to personalized, effective cancer treatments. I would like to thank Ms. Spinelli for mentoring me throughout this experiment and providing suggestions for this manuscript. Thank you.
Thank you.